speaker, we have Ms. Reena Clements. Ms. Clements is a PhD student studying neuroscience at OHSU. Her research focuses on how our sense of sight develops as we grow up. In addition to being an active member of the scientific community, Reena is also a senior communications fellow at OMSI and regularly presents her research in the form of an interactive demonstration to guest visitors. Please welcome our next speaker, Ms. Reena Clements.
Great, can everyone hear me? Yes. All right, so thank you again for the kind introduction and for the invitation to come speak today. So I'll be talking about something a little bit different, I think. So I'm going to share with you a little bit about my research, but not too much. Mostly I'll be sharing today my personal story about how I got interested in science and how that can translate to all of you who potentially are interested in science or pursuing a career in science. So this is me when I was just a little baby. From my early childhood, I actually really hated science. I thought I was going to be an author when I grew up. I was writing and reading at every chance I would get. This was to the great displeasure of my dad, who really, really likes science. He's not a scientist, but he was always encouraging me and my brother to do science experiments on the weekends. He would always go out and buy us model kits, and I didn't want to have anything to do with that. I would go hide somewhere, I would read a book, and that's just what I would do. That's what I thought I was going to be when I grew up. And that all changed when I started to take my 7th grade biology class when I was 12. So this is my dad, and this is me now. So in our unit on genetics, we learned about 2 types of traits, dominant traits and recessive traits. If somebody has 2 copies of the recessive trait, they will express that trait. If somebody has 1 dominant copy and 1 recessive copy, the dominant copy will actually mask the recessive copy, and you won't actually see that that person has that gene. So if you look here, I guess the laser's not working, at my dad's ear, his lobe is kind of attached to his face, whereas if you look at my ear, it's kind of got a little bit more of a lobe structure on it. So this is something called a free earlobe on me and an attached earlobe on my dad. Attached earlobes are a recessive trait, and free earlobes are a dominant trait. Because everybody has 2 copies of these, one that they get from mom and one that they get from dad, I knew that I had to have gotten 1 copy of the recessive gene from my dad, but a dominant copy from my mom because I expressed the dominant trait. So this was the first time that science was really tangible to me. I figured out something that I learned in a classroom that I applied to my real life. And I knew that this was something that I really, really wanted to do for the rest of my life. This, for me, was the spark that got me really interested in science. For me, this happened when I was 12. But this can happen at any point in anybody's lifetime. When I was in college, I frequently worked with elementary school students through a program called Wizards, where we would go out to elementary schools and perform science experiments with the kids whose schools didn't really have the resources to do any kind of science activities. And the way we would get them engaged would either be to make something really colorful or make something explode. This was the way that you get a child's attention. So here, I'm showing 2 different activities that were a favorite with the kids. On the left, we're making marbled paper using shaving cream and food coloring. And on the right, we're making marbled milk, where you put a little bit of soap and milk, a bunch of food coloring, and then you can touch it with a Q-tip, and it explodes into really bright colors. Today, I'll actually demonstrate another one of the activities that we did with the kids, which was a real favorite with them. So here I have a bottle of hydrogen peroxide that I'll put over here in this. I'll add some food coloring, because the kids really always like color. I'll add just a little bit of dish soap. And then pre-mixed here, I have a bowl of yeast. So when I add the yeast into this mixture, I make an explosion of a bunch of foam, and that is called elephant's toothpaste. So again, this was something that we used with the kids a lot to get them really excited about science, because you can add a bunch of things together and you get an explosion. But how do we bring this beyond, this is cool because it exploded? So I used yeast in my reaction as a catalyst to get the hydrogen peroxide to bubble up into this big foam. So all kids know that yeast is used in pizza and bread dough to make the dough rise. So we can ask these kids, well, how do you use yeast in your regular life? And how is that similar to what we did in the experiment with you here? And that gets them really asking questions and thinking, I can be a scientist too. Which gets me to my next topic about how people generally think about science. So there's two ways that we can think about this. We can think about this in a fixed mindset or a growth mindset. In the fixed mindset, someone thinks that they are born a certain way with a certain life path and they're only going to ever be good at one thing. Whereas in a growth mindset, we think that I can do anything if I put my mind to it, or I'm happy to make mistakes and learn from them, or I'm not good at this thing yet. I thought until I was 12 that I was going to be an author, and then I changed my mind. And I had to work really hard at some of the classes in science that were difficult for me to actually get to where I am today. And so I encourage all of you to go forward into science with a growth mindset that you can do anything if you put your mind to it. And this doesn't just apply to kids, this can apply to any adult who is interested in learning about something new. So after I decided I was really interested in biology, I wasn't sure where to go with that. So one of the things that I did was actually a summer program for advanced high school students in New Jersey, where we had opportunities to take things that were outside the normal realm of things that we learned in our science classes in high school. So one of the things I did was I got to dissect a lamprey, and I learned about its unique digestive system as a parasite, about how it digests blood from the animals that it latches onto. I also got to stick electrodes all over myself to learn about how my body works in terms of reflexes. And I got to stick my head in a bucket of ice-cold water to learn about how my heart rate changes when my body temperature is different, for example, exposed to a warmer environment or a colder environment. I also did things like take a mini medical school course offered by one of the local hospitals. So here I have some notes about heart transplantation. And at that program that I was discussing earlier, I also took a physics class. Because, you know, it's science too, so why not try something new and take a physics class? So if these kinds of opportunities are available to you to take things which are really outside the normal realm of what you learn in school, I highly encourage all of you to seek out those kinds of opportunities and explore different things. Figure out what you like, what you don't like, and what really, really makes you excited about science. So none of this changed when I went to college. When I was in college, I changed my major a lot of times before I figured out what I wanted to do. So I was interested in biology. For a while, I thought I was going to be a marine scientist. So I learned how to scuba dive, and I took a trip to Belize to do research on coral reefs. I also had a summer internship where I studied the behavior and genetics of fiddler crabs. I had another internship in bioinformatics to try to figure out how genes correspond with different diseases. I took an advanced math class where most of the time we were sitting around a conference table doing math problems on Friday night for fun. And then finally I settled back on neuroscience. So here is a picture of me looking at how neurons in the part of the brain that help us feel things, so the touch, talk to other parts of the brain. So after changing my mind a lot and trying a lot of different things in biology and in physics, and as you'll see in chemistry, I finally figured out what I was really passionate about and what I wanted to do. So I'd like to bring this back to the summer program that I did when I was in high school for advanced students in New Jersey. This was my team project. So as part of that program, we not only took classes and things like physics, but we also did a team research project. There were some available in biology, but I decided to be different and I wanted to try something in chemistry. So this is the group of us that did that work in chemistry. We were trying to figure out how to coat things, such as in Teflon, to make better nonstick coatings. So of this group, Matt, who's in the white shirt in the middle, is the only person who's actually still doing chemistry. I've labeled a couple of my other teammates who are doing things like running her own business to be an SAT tutor. We have 2 engineers, 2 different kinds of engineers. One person is in medical school, 2 people are studying economics for firms in New York, and then I'm getting my PhD in neuroscience. So it was really great for me to have this opportunity to be able to explore, ask questions, and figure out what a lot of the different things were that I liked and what I didn't like, have the ability to ask questions, make mistakes, and go forth for being really, really excited about science. So, funnily enough, this is some of the work that I do now. So I use complex genetics to study how the visual system develops. So that means when you're growing up, how do those cells in your eye tell your brain what you're seeing? And how does that go wrong in disease where some people can't see because they have problems when they're initially developing as a child? Those connections don't form properly. So here I have an example of one of the things I do. I have listed the genotypes, that means what genes they have, of 2 parents, and what possible kinds of offspring they can have. So 3 of these will come up with a disease, and the rest of them will be normal. Which is actually kind of interesting that that's initially what I was interested in 7th grade, and then I changed my mind a whole bunch of times, and I wound up sort of exactly in the same place. You might be looking at this slide and thinking, that's really complicated, I could never understand that. Well, another thing I do is that I'm a science communication fellowship at OMSI, which means that I go to the museum and I've built an interactive demo that I share with visitors of all ages from really, really young to older folks. And so if you look here, I have these 2 buckets with eyes on them, and then I have some tubes crossing in the front. So the kids or any museum visitor can take some marbles, put them in those buckets. Those represent the eyes, the marbles represent the neurons, and we see where they go in the brain, which is below the picture here. In the normal condition, in our eyes, input from our right eye actually travels to the left side of our brain, and input from the left side of our left eye actually goes to the right side of our brain. So when we drop the marbles in these buckets, they'll travel through these tubes and cross to the opposite side of the brain. But what happens in a disease state, like I mentioned previously? Well, I flip a switch, and if you'll see, there's a bunch of nails behind those tubes. The marbles, instead of going into those nice tubes, will travel through those nails, get mixed up randomly, and drop into the buckets in the wrong side of the brain, which is actually what happens in the model of the disease that I study. So earlier I did a little experiment to show what I did to get kids interested in science. So I'm going to repeat that experiment, but do it a little bit differently. Another difference that I think we can make, one of the biggest differences, is actually going out into the community and helping people become interested in science. I talked about the fixed mindset and the growth mindset. In today's society, a lot of times we discourage people from entering science. You'll see shirts at, high companies that say, oh, I'm bad at math, and girls will wear those around. We want to discourage that because that's a fixed mindset. Anybody can do science if they put their mind to it and they ask the right questions. So I'll repeat the experiment to show how we can be a catalyst for getting people interested in science. The agent that I used before was 3% hydrogen peroxide. I'm going to repeat that experiment with 30% hydrogen peroxide. So this is sort of a representation of if we all work together, go out into our community to spread the love of science, what we can actually do. So this one is a little bit more dangerous. So I'll be putting on my PPE for this and moving a couple of things off of this cart. I'll leave this one up here just to show you the difference in what it's going to look like. So again, my dish soap. Food coloring. 30% hydrogen peroxide. And finally, my catalyst. So not yeast, but something a little bit different. So this is what happens when we use more of something to, in my analogy, get people excited about science. It'll go on for a while.
What was your catalyst?
Potassium iodide. So this is actually just oxygen and soap and steam, so it's not dangerous. And it's still going. So this is what we want to do in our communities of science. This is how much excitement we want. I'll take any questions if anybody has any otherwise. Yes?
You talked about a fixed mindset and a growth mindset.
Yeah.
Besides exploring things, how can we get more kids interested in STEM and science?
So for littler kids, I usually make things explode and make things colorful. For more middle school age kids, I will try to relate it to something that they know already. So for example, a lot of middle schoolers will come to me and say, well, my parent or my grandparent or my family member had this disease. What can you tell me about that? And that gets them really interested. In high school, a lot of times it's just motivating to tell them, hey, these are some different careers that you can have. And this is why they're interesting. At the college level, a lot of people will say, I'm just taking this class because it's required. But I will relate that back to something that they already know about. To sort of get them to realize, like, hey, this is actually really, really interesting. And for adults, it could just be as simple as a visit to the science museum to try to learn something new or to talk to somebody who they know in science and really talk to them at a level that they can understand. Because if a scientist talks to you, and if I had told you what that slide meant of all of those possible combinations in offspring in my parents, you probably would have tuned me out. But if I bring this kind of demonstration to a museum, this is actually what my research is on. And a 4-year-old can understand that. So really just relating to whoever you're talking to and making sure that they understand and you're not talking in too jargony of language. Mm-hmm. So I'm not a chemist. For some reason, the potassium iodide does not work as well in the little one, and using yeast as a catalyst does not work as well in the big one. So I'm not sure what exactly it is that makes it explodes so big because I used a higher concentration of hydrogen peroxide.